Replication is the process in which DNA copies itself. This takes place during cell division. And the result is an ideally identical DNA molecule. When DNA undergoes replication, the two DNA molecules that are produced each contain one original strand of DNA. So let's draw that with a picture. Let's say that we have a double helix, which I'm going to draw in blue. So there's one strand and then there's the other strand. So here's our very poorly drawn double helix, and this undergoes replication. Those two original strands of DNA are separated from each other, and each one ends up in one of the resulting DNA molecules. The other strand is a new strand. So how can we phrase this? After replication, each DNA molecule contains one old strand and one new strand. The old strands in this diagram are in blue, and the new strand is in red. The new strands, as they are synthesized in the same direction. Both of the new strands. And they're also synthesized at the same time. So they're made at the same time and in the same direction. The direction that they're synthesized in is starting at the 3' end and moving to the 5' end. However, because our two strands of DNA are one is running 3 to 5 and the other is running 5 to 3, in order for both strands to be synthesized simultaneously in the 3' to 5' direction, they can't be synthesized in the exact same way. So let's say that the 3' end is on top for the red strand and 5' end is on top for the, I said that backwards. 3' is on top for the blue, 5' is on top for the red. So the blue strand can be synthesized in this direction, like we would like to see, like that, no problem. But the red strand needs to be synthesized in the opposite direction. So the red strand needs to be synthesized from its 3' end to its 5' end. The synthesis of the DNA strands happens at the same place at the same time. So one can't start at the top and the other start at the bottom like that. It won't work like that. They have to be synthesizing in the same general area at the same time, which means that one strand is being copied in one direction and the other strand is being copied in the other direction. And this creates a really interesting, complex situation. Let's look at a picture from your textbook that really, I feel like, shows this really well. This is figure 22.6. And so this is showing us the strand of DNA, and it has so in this diagram, the two strands of DNA have been separated from each other right here so that they can be copied. And one of these strands, the blue, let's see, the strand that is right here, this particular strand, is being synthesized from its 3' end up. So it's being synthesized in this direction. This red strand that's being synthesized, or the red strand that's shown here, is the new one that's being formed. The other blue strand, so this strand right here, is being synthesized in short segments in the 3 to 5 direction. And those short segments are eventually going to get linked together to make one large strand. But because this other portion is not allowed to be synthesized in this direction, it can't be synthesized consecutively or consistently. So the DNA will open itself up a little bit, and a small fragment will be synthesized, and then it will open itself up a bit more, and another fragment will be synthesized and connected, and then it will open itself up a bit more, and that process will carry on. So replicating this side of the DNA is quite a bit more tedious than replicating the other side. The strand that is able to be replicated continuously, the one that's, pointing in the right direction for this to work well, is called the leading strand. The leading strand is synthesized continuously or copied continuously from the 3' to 5' direction. The other strand that has to be made in fragments is called the lagging strand. And that is the piece that is synthesized or made or copied in fragments, which are going to eventually get connected into a continuous strand.